3 years ago, I concluded a very impactful academic research study where I answered the question, how did Cowessess First Nation develop and implement its Mi'opimatiswin Act to retake control over its children? I found that years after the implementation of the Act, not a single Cowessess child was apprehended and placed into government child welfare. I had the opportunity to disseminate my research to thousands of people all over Canada and beyond. Amazing research is being conducted by racialized faculty and graduate students all over Canada in fields from biology to psychology, from public policy to public health, from research in film to research in education. Racialized researchers are doing amazing work to solve real-life problems. And now they're here to share it with you in very simple terms. My name is Kwaku Ayisi, and this is Academic Research Made Simple. Hello everyone, welcome to Academic Research Made Simple podcast, or ARMS. This podcast is sponsored by the Digital Research Alliance of Canada. My name is Kwaku Ayisi. I am a PhD candidate at the Johnson Choyama Graduate School of Public Policy here at the University of Regina. Incredible research is being conducted by racialized graduate students and faculty from British Columbia all the way to Nova Scotia and everywhere in between in Canada. Today, we have an excellent researcher, Michael Mensah, as a guest on this podcast. Michael is originally from Ghana, where he spent his formative years near the beach, a setting that fostered his natural affinity for water and its related studies. Michael earned a BSc in Natural Resource Management, specializing in fisheries and watershed management from Kwame Nkrumah University of Science and Technology, Ghana. He later received the prestigious Commonwealth Shared Scholarship to pursue an MSc in Integrated Management of Freshwater Environments at Queen Mary, University of London, UK. Currently, Michael is a PhD candidate in biology at the University of Regina, funded by merit-based awards from the Ministry of Agriculture and the Saskatchewan Cattlemen's Association. He works under the supervision of Dr. Kerry Finlay. Michael is passionate about working with natural processes to enhance water quality. His research focuses on predicting salt dynamics and identifying effective strategies to keep pond water fresh for cows and other livestock. Outside of his academic pursuits, Michael enjoys playing and watching sports and volunteers at a soccer development center. Most recently, he achieved national recognition by winning the 2024 Three Minute Thesis competition after excelling at the local, regional, and national levels. Michael made history by being the first student at the University of Regina to win the National 3MT competition. It's great to have you here with us today, Michael.
Thank you. Thanks for having me.
Great. Can you tell us a bit about the pathway to you becoming a PhD candidate, and specifically in biology?
Yeah, that's an interesting question that I kind of smile at whenever I'm asked. So like you rightly mentioned earlier before, that I grew up by the beach. And yeah, I always like to think that created me a natural affinity to water and its related studies. However, I would really trace this path to happenings during my undergraduate studies. So during my undergrad, I read natural resources management and majored in fisheries and watershed management. And as part of the things that we did there, we had an aquaculture demonstration farm where we had practical sessions of what we learned in class. So we had fishes in these ponds, and there were lots of research going on at the time. Some monitoring live species, others monitoring water quality. for my undergraduate dissertation, I had the opportunity to work with prawns. So specifically, I worked with Macrobrachum volenhovenine. So they are basically freshwater prawns in Ghana. So at the time, we were looking at opportunities of diversifying culture species in Ghana, which is mostly centered around tilapia and catfish. So during this period, I had the opportunity to work with live species, and also, so it was something very novel because we were trying to fine tune culture practices for freshwater prawns, as I said, trying to diversify cultured species. It was very challenging. And while the primary goal of raising these species from larval stage to a stage where they could be raised was unsuccessful, we managed to make a lot of headway in regards to what salinity level or what levels of salt in their water enhanced growth to a particular stage. So during this period, something very significant happened. Significant in the sense that I would say that sort of informed the direction of my studies. At the time, I became so attached to these live species. They felt more like babies to me. And you go to the lab, with high hopes that, you have adjusted a few things that didn't work before and hoping that these species will survive. And you come back to the lab the next day and they are gone and you're so sad. And I'll tell you a funny thing that happened. So I visited the lab with a friend one time over the weekend to check on these species. And unfortunately, the gravid ones are quote unquote the mothers, the pregnant ones that bear the eggs, was dead. And all I could think of doing at the time was, discard it, because I thought of them as my babies, and this friend is like, can't we eat this? You buy from the market and they are there, this is there, you haven't put in any chemicals that doesn't make them edible or whatever. I'm like, oh, okay. So these are actually edible. I had discarded lots of it just because if they didn't succeed at, growing to the stage I wanted, it felt like a loss and had to be discarded. So at that point, I got to realize that I was pretty much emotionally attached working with live species. So I made the decision, okay, so I am either working on live species or the water they live in. So if I could, make the environment they live in more habitable for them, then it would be something that I'm doing for animals that I care about. So life in water. So I decided, okay, then I will focus more on water now than the live species in water. So that series of events sort of shaped the direction that I wanted to go with my interest in biology. So fortunately, after my undergrad, I secured 2 positions, actually, one to study at Queen Mary, where I ended up, and there was another to study sustainable aquaculture at the University of Stirling, also in the UK. But based on the premise that I earlier talked about, I'm like, sustainable aquaculture, I might be doing something more live related. So I'm like, okay, integrated management of freshwater environments sounds like, yes, I am still into aquatic sciences, but, dealing with water rather than live species. And that's something that I like the idea of doing that. So decided to pursue that in the UK. And as a natural progression from there, I got to know about my supervisor that I'm currently working with, Dr. Kerry Finley. So as part of what I did in the UK for my dissertation, I worked on assessing water quality improvements that some citizen science activities had delivered to canals in London, specifically the Regent Canal. So as a natural progression from there, I got to know that my supervisor is also interested in projects that make use of working with natural processes to improve water quality, which is what I found interesting doing. So, talking about my decision to pursue higher studies in the field that I am pursuing now, I think this is the background that I would lay.
Awesome. Yeah. So it sounds like water quality has been your main motivator.
Yes.
Throughout.
Yes.
Can you tell us a bit, for those who are not familiar with what biology is about? And I know the scope is very, very broad, but specifically regarding some of the different research that is being conducted within the Department of Biology, either here at the University of Regina or just generally, in case somebody is wondering, what do these people who call themselves biology experts do anyway? What are some of the different researches that are being done, and how could it be impacting different groups of people?
So biology, in its basic definition, I would say has to do with the study of life and the essential processes that drive nature. That's how I broadly explain what biology is. And within biology, there are several fields, I would say, within biology that you can focus on. So talking of fields, we've got a molecular biology. So the type of biology that focuses much on, the basic units of life, talking of cells and DNA, RNA and stuff like that. We've got the environmental biology, which is my field of study, I would say. So we try to understand processes that happen in the environment in general. So what's happening in the landscape and how is it affecting life in water, what's happening in water, and how is it happening in the lands that surround it or the atmosphere above it. So stuff like that. There is ecology that has to do with the study of life within water. So animals or fishes and their interactions with each other and with the environment that they find themselves. And I think more recently, there is also the field of bioinformatics, where, biological data is, being assessed with some rigorous mathematical procedures and stuff like that in, helping to understand diseases better and, coming up with medicines and stuff like that. So, biology, I would say, is broad, and like I just described, there are several interesting fields within it and interesting research currently happening. And specifically here at the Department of Biology here at the University of Regina, it's a broad field that cuts across. So my lab, for instance, I mentioned we are into lots of environmental biological research. So I've got lab mates working on greenhouse gas emissions from water bodies. So greenhouse gas basically drive what we call global warming and by extension climate change. So these gases, when they are released into the atmosphere, have the characteristics of impacting how warm the globe gets. So I've got colleagues also working on understanding toxins within aquatic systems and what triggers their release and stuff like that. So specifically, I've got a colleague working on microsystems, which is a toxic and we try to understand what drives algal blooms and all of that. Other interesting projects happening in the fields of ecology. There are people looking into how fishes interact within their environment, within their environment affects their survival, reproduction, and general fitness. There are researches into microbial communities, so microbiology, microbial communities, talking about bacteria, fungi, that we can't see with our naked eyes. We need the help of microscopes to see. So there are lots of really interesting projects going on. And these fields within biology are well represented, I would say, here.
Okay, awesome. Yeah, very broad scope of very incredible work being done in the biology department.
Yes.
Your research topic, drivers and controls of salinization in prairie ponds. If you were explaining this topic, just the topic and not going into the weeds to, let's say, a 10-year-old, how would you tell them what it is that you're attempting to do?
Okay, so I am attempting to understand what causes prairie ponds, what causes an increase or decrease in the level of salt within prairie ponds. And by prairie ponds, it's a term that collectively describes wetlands and dugouts. So wetlands are naturally occurring water bodies. Basically, in one of my 3MT showcases, one person working on wetlands described them as nature's kidney. And what does kidney do? Kidney basically filters pollutants from the bloodstream so that whatever is circulating within your system is pure enough to enhance a healthy living. So wetlands basically are naturally inundated areas that help to filter lots of runoffs. So runoffs, water that come from agricultural activities. So when it rains, water just doesn't stay or leach into the soil. There are instances where it runs across and they move in a direction and typically end up in waterways, talk of lakes. And typically these runoffs go through wetlands where they are slowed down for a while and then purified, as it were, before their eventual discharge into our natural waterways. So wetlands are very important natural bodies of water. And here in the prairies, they are sometimes utilized as source of water for cow watering. So that's wetlands. And I mentioned dugouts. So dugouts are artificially constructed water bodies. And they are constructed to be typically deeper than the wetlands, just so they typify their primary roles as water storage units. So, in my study, I collectively term wetlands and dugouts as prairie ponds. So, drivers and controls of salinization in prairie ponds basically is looking into understanding what it is within the environment that surround prairie ponds that influence how salty or how fresh these ponds stay.
Okay, awesome. So for those who are tuning in from outside of Canada, can you describe or explain what are prairies?
Okay, that's a good question. So the prairie regions of North America are actually regions of vast grasslands. Okay, so the prairies typically do not have trees growing here naturally. So though we have shrubs, which are relatively shorter and non-woody trees, we don't typically have the timbers like I would expect from the part of the world that I am originally from, where we have the rainforest and all of that. The prairies doesn't naturally have that. The prairies also receives less amount of precipitation. By precipitation, I mean rainfall and snowfall. So compared to the rest of the regions within even Canada or North America, the prairies typically receive less amounts of precipitation. This is one of the reasons why farmers will typically want to create dugouts so that they are able to store water from the little precipitation that they receive, so that they are able to water the cows that they raise here. And cow production in the prairies is a huge industry. So basically, that's how I would describe the prairies. It's a semi-arid regions within the northern Great Plains.
Okay. What is the main issue in the research that you're doing? What is the main problem that you're trying to find out more information about and ultimately make recommendations to make better?
Yeah, so the main issue here is I talk about salinization, I talk about saltiness throughout. The prairies is a region that is naturally salt rich. And I have been using the term salt quite broadly and loosely, but getting into a little bit of detail, the ion that I am most interested in is sulfate. So sulfate is a type of salt, okay? And high levels of sulfate in water is pretty toxic. So much so that typically it is recommended that you keep sulfate level, like ponds that have sulfate levels less than 1,000 milligram per liter is what's usually ideal and recommended for cow watering. Between 1,000 and 2,000 is kind of acceptable, but beyond that is really considered not ideal and not recommended for use in cattle watering. So my research is basically looking into how sulfate levels can be kept low within these ponds. The problem here is that, like I mentioned earlier, the prairies is generally sulfur rich. And when these ponds, especially the dugouts, are created, they may provide fresh water for the first couple of years that they are created. However, as it progresses, they tend to get salty over time. And salty water or dugout water that has high sulfate level is not ideal and is unsuitable for use. So the problem here is that these dugouts get salty over time, and then we are trying, and that makes them unsuitable for use. So we are trying to keep them fresh enough for cattle use. That's what we are looking into. So
you're mostly looking at cattle as the end users. So this has nothing to do with human consumption.
No.
So
cattle is the focus, but typically farms have other livestock that they keep. So it would benefit those ones too if it happens that a farm has, bisons or any other livestock that you can think of. Yes.
Okay, awesome. What are some of the findings? So what are you seeing in available literature on research or similar research that has been done regarding salinization in ponds, maybe in a different province or different country, and what is the literature saying?
Okay, it's a good question. Talking of
Either in terms of how they define the problem or in terms of mitigation efforts that they might be using in a different jurisdiction or a different place.
okay, in literature, like I said, it's established that the prairies that we find ourselves are is sulfur rich, and it's kind of patchy. Like it's not like everywhere you go or every spot you step on, you would find the soil to be at the same level of salt enrichment. So it's kind of patchy where this occurs. So while the prairies in general is considered to be sulfur rich, it is also established that there are areas that are considered discharge areas. So discharge areas basically are areas where there's sort of groundwater release into topsoil. So in these areas, you typically find the soils to be more salt rich. Just because groundwater discharge, what they tend to do is to dissolve whatever ions are present in the earth, and then transport them as they travel up to topsoil and then release them there. So discharge areas are typically salt rich, whereas there are recharge areas. So recharge areas are areas that tend to allow the leakage of salt, I would say, into the earth. So recharge areas are typically less salt rich, and discharge areas are typically more salt rich. So so generally, there's literature recommending, trying to position these dugouts at appropriate places and not making sure that you are not, situating a dugout in a discharge area where it is more likely to get salty quickly within a short time. Whereas people are encouraged to site their dugouts in recharge areas where you are likely to have your ponds hold fresh water for a longer time. The issue of salinization isn't one that kind of cuts across a wide area across the globe, because there are some areas that don't have to deal with high levels of this in their ponds and all of that. And even within North America, I've been to conferences and several academic events, and I have heard other researchers talk about salinization, but their focus is different. So there are those that are focusing on sodium chloride and other basic salts that are used on our roads, especially during the winter season. So you go to other parts. I don't think we have much of that in Saskatchewan, but you go to other parts of Canada and even North America. And then when it rains, typically, rather than using sunlight, we typically do here on our roads, it's salts that they use because it helps melt it and, help with traction of vehicles and stuff like that. So, and these are salts that get washed and eventually discharged into lakes and natural waterways. So there are other research going on trying to investigate into different types of salts. So those are the ones. Talking of mitigation, there are studies, especially in Asia, that have reported on using artificial floating islands to improve water quality. Mostly, they've been used to reduce levels of nitrates and phosphorus, phosphate. So not many have really looked into using it as a means of mitigating sulfate levels in water bodies. So when it comes specifically to using these artificial islands as a means of mitigating salinization as a result of high sulfate levels, not much is currently available. There are a few that have come out from other North American regions. I think there's one that I chanced on from the US that also used it, but in a mesocosm setting. So mesocosm is when you create something to simulate natural environment. So in a tank or something to investigate what will happen in a natural setting. But for one that is investigating the effectiveness of islands in natural settings, I haven't come across yet. So that's something novel to what we are doing.
Okay. So your objective for the study would include, would it include testing to see if some of the mitigation being done elsewhere would be applicable?
Yeah, that's one thing that we are doing with the floating islands. There's another mitigation strategy that I am testing that is dredging. This is one that I think we farmers in the prairies do and have done in the past, but it's more of we think we want to do this, and I haven't found much empirical procedures being employed to understand what's really happening. So one of the things that I am doing is we have a couple of ponds that were dredged, and we are using empirical procedures to test how effective dredging actually is at improving water quality in these ponds.
Okay.
Yeah.
Okay. You might have touched on this already a bit in terms of your methodology or how you're going about in answering your research questions. Can you break that down a little bit for a layperson,
right?
In non-technical terms, how you are answering the research questions that you have come up with?
Okay. So in answering my research questions, there are surveys that I undertake out in the field, and I collect samples. I collect water samples and I collect soil samples. I also do collect sediment samples. So the difference between soil and sediment in my research is that soil samples are actually soils within the uplands, so the areas that surround the ponds that I work on. And underneath, these are natural or earthen ponds. So within the inundated area, there is also soil at the bottom. So I collect samples at the bottom of these ponds. I also do collect water samples. In the field, I collect what I describe as limnological variables. So there is this fancy equipment we have where that in the field, we call it the YSI. We just drop it in the water and it's able to tell us a lot of things about a water body. So it's able to give us an idea how salty it is, how much oxygen there is, pH and stuff like that. So I go out to the field and take all these records and also come back with these samples that I just talked about. One other thing that I am doing is I complete what I call the pondscape survey. So I go out to the field and I have designed something that allows me to take records of what's happening within the environments that surround the pond. So how vegetated is the environment that's around this pond, what type of crop there is, what kind of land use activities are happening around this pond, details like that. So with details like that, I come back to the office. What I am doing also is having collected the water, soil, and sediment samples, I come back to the lab and assess them for several variables.
Okay.
And then once I have all this information, what I am hoping to do is to sit down and analyze them to identify patterns. So what are the variables that correlate to low sulfate in pond water, and what are the variables that correlate with high sulfate in pond water? And in doing this, I am hoping to arrive at identifying what's driving or enhancing saltiness in this pond and what's controlling or mitigating salt levels in these ponds.
Awesome. Sounds very, very interesting.
It is.
Yeah. Have you identified, or do you perceive any limitations in the research that you're doing?
Good question. I would have to think about that for a second.
Okay. Okay. I'm guessing it's probably where you are in your research, because usually after towards the latter part of one's research, they might begin to actually think about what some of the limitations are. Some maybe at the beginning, but I'm guessing maybe it depends on where you are.
Talking about if you put it that way, I think I must have identified a few of them already.
Okay.
So taking records of precipitation, how much snow or water the areas where my ponds are situated receive, isn't something that I paid much attention to. And I'm at the point where I feel like, I wish I did, because I talked earlier about discharge and recharge areas. There is a way to sort of more accurately measure them with what we call piezometer data. And that's a fancy way of measuring it that I didn't have for my project. But I believe that, records from precipitation would have made it easier to identify it. However, all hope is not lost. There are other procedures we are trying to use, albeit more complicated or not as straightforward as these might have been. We are still hoping to be able to characterize them into recharge and discharge areas. That's something that I would have considered if I had had as much thoughts as I have had.
Okay.
Yeah.
Okay. Have you gotten to the point in your research where you've come up with any significant findings, or you're still getting towards that part?
I'm still getting towards that, and to that point where I would make very conclusive claims. However, there are a few consistent patterns that I have seen already, and that I can talk about. For instance, do not situate your ponds at regions where the soil is salty or salty to a certain degree, because it would eventually end up in your pond. So that's something to consider. Also, what's proving to be consistent is, vegetated landscapes around these ponds. So if you have much more vegetated landscapes around your pond with deep-rooted vegetation, then you are likely going to keep your pond fresh for a longer period. So and talking of vegetation, there are areas where the cows being raised have been given direct access to these ponds, and what they do is that they tend to trample on these vegetations, so eventually these areas that surround the ponds are unvegetated and bare, and that sort of allows much more transport of these salt ions from the areas that surround them into the pond. And aside from that, I am also finding that, so I talked about collecting samples from the sediment, soil, and water. One of the things I have done so far is to look into the sulfate levels in these three regions and to understand the relative importance of different inputs. And one of the observations that I have made is that salt is typically highest, like at any point in time, it is typically highest in the sediment. So if you're allowing direct cow access, then essentially the trampling and all of that will cause the release of these ions that would otherwise be buried in the sediment to be released into your water where you don't want them. So yeah, these are a few things that could be done to hopefully increase the lifespan of these ponds.
Okay. So basically, the salt levels in pond water has to be reduced for the cattle.
For sure, yeah, because high salt levels, I don't know if I touched on this already, but when water is salty, cows are able to tell, and they will either drink less of it in the event where there are no other alternatives. And if they keep drinking and it gets to that level where it's unbearable, they might stop drinking altogether. What that does to you as a farmer is that production will be impacted. So if they are dairy or produce milk, then they are not producing as much milk as you want. If you are growing them for the meat, they are not putting on as much flesh as you'd expect. So their health are impacted, and then the effort you are putting in as a farmer is not yielding the results you're expecting.
Okay, awesome. Well, let's switch gears a little bit. Now, the next question you can answer either as personal experience or generally what you've seen. As a racialized researcher, are there any barriers to success, opportunities to success, or perceptions, ideas, worldviews that you find are brought to academic research either by yourself or by other racialized researchers? So barriers, opportunities, and perceptions as a racialized researcher.
That's a good one. I think I would address this question from a personal perspective. Personally, I tend to draw lots of inspiration from people that have done something that I hope to do. And typically, I would want to draw lots of inspiration from people that, actually look like me if they have ever done it. I think that what could be some sort of a barrier might be, the gap in representation. I see this, and on a practical sense, this whole earlier on, you talked about the national recognition I received in regards to 3MT. The first time I actually saw you, a video of you as the U of R 3MT champion for 2022, I was really motivated. And I felt like, oh, great, that's quite cool. A guy that looks like me. And he went all the way to do this. This is something I could do too. So it was so, I believe that if, you have dreams and thoughts of doing something that you look up and, all you see is people that don't look like you that are able to do that. That is not to say that you should shy away from a dream that you hold if you have not seen people who look like you do it before. That should actually be another motivation that drives you to do it, so that generations that would come after you can look up to see that motivation or inspiration that you were hoping to see. So, but on a personal note, I draw huge motivation from that. So I believe that if the gap in representation could be addressed for a lot of the fields, that would be great for racialized groups or students.
Okay.
Talk of opportunities. I mean, like I also said in this representation, if you look up there and you want to achieve something that you don't see someone look like you in the list of those that have done it before, then that's an opportunity to be the first person that looks like you to ever do it, right? So that's how I think of the opportunities too. But I think, that's how I would like to address that question.
Okay, awesome. Regarding disseminating your research to a broader non-technical audience, how important is it? And in answering that, specifically for you, who did that on such a high level and did excellently at it in 3MT, how can you describe what 3MT is for those who don't know it and its advantages?
Okay. So 3MT generally is, a term for, it's an acronym for 3 Minute Thesis, and this is an event that challenges grad students to distill their information into easily digestible and accessible forms to a broader audience or a general audience. I find it particularly useful in the sense that the 3MT actually provides the opportunity to bridge the gap between academic research and the broader communities whose lives we try to improve with our research findings. Because typically, people think of the university as, this area with four walls and whatever happens in there stays in there. But no, the university really is, an institution with lots of, people who are committed to advancing knowledge. So what we are really doing in here is, looking into issues of social interest, whatever field you find yourself. I find myself in the field of biology and, researching into this very important issue in environmental biology. And, there are amazing research happening in psychology and other departments and all of that. So basically what we are doing here is looking into things or issues that are of social relevance, and whatever we find here do not only impact our lives, but impact the broader community out there. And I think it's important that we are able to communicate our findings in languages that people that whose life we are hoping to improve can understand. So I think that 3MT provides the rare and very prestigious opportunity to disseminate your findings to these people that you care about.
Right, absolutely.
Yeah.
Okay, are there any people who have inspired you across the years to conduct academic research at a PhD level or even higher? Do you have any mentors, role models, or people that you looked up to that have motivated you to where you are right now as a PhD researcher?
Right. Yeah. Specifically to academics, growing up, there are lots of people that, have played huge roles in our lives. And yeah, we've looked up to them, and their values have shaped where I find myself now. But specifically to uh, my interest in pursuing a PhD and eventually, hopefully, an academic research-based career, I think I would think of two professors during my undergrad that I had the opportunity to closely work with, Dr. A.J. Boateng and Dr. Kwesi Obrikrine. Dr. A.J. was my supervisor during my national service, which is an obligatory 1-year service that all people that complete higher education, so post-secondary education, actually have to serve. And I served as a teaching assistant. In Ghana. Right. I served as a teaching assistant at Kwame Nkrumah University of Science and Technology, where I completed, and he was my supervisor. Okay. And during this period, I had the opportunity to work closely with him on some projects that he was working on. And I would say I drew lots of inspiration from what he does. And he was essentially a mentor. And I enjoyed working with him on all the projects that we worked on. So that's something. And thankfully, he's also into aquaculture. However, during that period, we worked on lots of water quality research. So we worked on lagoons. We did stuff like that. So when I was transitioning from live animals into water quality research, like he gave me that feel, and I felt like this is something I want to do to a higher level. Also, Dr. Obrikrine, worked closely with him on some environmental impact assessment project. So I remember traveling with him on some fieldwork activities to sort of assess and establish baseline conditions for some streams and water bodies prior to the mining of a concession close to these areas and stuff like that. So these are water quality research opportunities that I had the opportunity to work on based on, these two amazing mentors giving me the opportunity to be on these projects. Yeah.
Okay, wonderful. And now the last question is on the back of this one. Just as you had mentors or people you looked up to, I'm sure there have been people in the past, there are people currently, and there'll be more in the future who'll be looking up to you and taking lessons from your journey or your status as an academic researcher. If you had any message to give out there to maybe a high school student who is weighing their options on whether to become a researcher and pursue a PhD just like you, or even an undergraduate student, or even somebody who's been out of school for a long time and has come back and is considering charting the same path, in a very few words, what sort of advice would you give to them?
Oh, I feel like that's a hard one, but I mean, in few words, I would say that dream and then have the passion to pursue it, right? This is a level that years back I dreamt of, and I dreamt of being, and I am now. For sure, it hasn't been a straight line or a smooth path to chart, but I believe that the resilience to continue and the passion to pursue have really influenced, activities that I have done in the past and still continue to do so. So you've got to dream and have an idea where you want to go, and then you've got to pursue that, because definitely, dreaming and having an idea where you want to go, and then you've got to pursue that, because definitely, dreaming and having an idea where you want to go, and then you've got to pursue that, because definitely, dreaming and having an idea where you want to go, and then you've got to pursue that, because definitely, that there would be roadblocks that you would have to get around. And yeah.
Wonderful. Wonderful. This has been a great discussion with you about your research work, about your research journey. And I really appreciate you coming and sharing all of that with us. Thank you very much, Michael.
Thank you. Thank you.
Thanks for tuning in to Academic Research Made Simple podcast, or ARMS, sponsored by the Digital Research Alliance of Canada. Tune in again to hear more incredible research being done by racialized graduate students and racialized faculty here at the University of Regina.